Hello everyone. In this session we are going to discuss about the chapter 1.3, that is digestion, of the paper Physiology, Biochemistry and Immunology. So in the previous classes we have already discussed about the gross anatomy of human digestive system. So now mainly we'll discuss about the digestive glands. So already we have completed the organs. So now mainly we will concentrate on digestive glands. So what are these digestive glands? So these digestive glands are associated with the digestive system. So mainly there are 5 different types of digestive glands. One is salivary glands, liver, pancreas, gastric glands and intestinal glands. So let's see in detail about the first one, that is salivary glands. So there are 3 types of salivary glands, that is parietal salivary glands, submandibular salivary glands and sublingual salivary glands. So as you can see in this picture, there are 3 different types of salivary glands. So the first one, parietal glands, there is only one pair and they are the largest salivary gland below the ear pinna. So there you can find the Stenson's duct which arise from each gland, and there is the second molar teeth, upper jaw and the cheek. So in that region it opens up, and these parietal glands secrete enzyme, and if the viral infection takes place to these parietal glands, that leads to mumps disease. So that is also called as, or it is caused by paramyxovirus. So that is about the parietal gland. So second we'll see about the submandibular. So you can find out the submandibular mandibular gland. It is mainly present near to the neck region. So submandibular, it is only one pair, and lower and it is mainly the junction between lower and upper jaw near to the cheek region. And Wharton duct, they arise from each gland and open up to the lower gland. So that is about the submaxillary or submandibular glands. Next we are having sublingual. So lingual, they are related to the tongue. So you can see sublingual, they are mainly present on the tongue and near to the tongue regions. So sublingual, they are also one pair, and they are mainly present in buccopharyngeal cavity. So buccal means mouth, pharyngeal means near to the pharynx, and there are 6 to 8 ducts which arise from them, and these ducts, they are called as ducts of Ranvier, and they open up near to the tongue region at the buccopharyngeal cavity. So these are the 3 different types of salivary glands which are associated with the digestive glands. Okay, so next, what does the saliva secretes or salivary juice means? So the secretion of salivary glands, that is called as saliva or salivary juice. So by explaining this juice only, I am going to tell about what is the function of this and how the digestion also takes place along with these glands. So daily it is secreted around 1 to 1.5 liter per day, and chemical nature is slightly acidic. pH, it is 6.3 to 6.8, and the control of secretion is autonomic reflex, means, for example, autonomic means automatic. For example, if I keep a bowl of sweets in front of you, so you automatically salivate. So it is not controlled by us. So that is autonomic reflex. And chemical composition, water is around 99.5%, mucus, salts are present, and enzyme. So what type of enzyme? Salivary enzymes. So that is phthalate and lysozyme. So the functions of salivary juice and its enzyme is to, saliva makes that medium slightly acidic and the action of the enzyme starts. This also helps in taste, deglutition, speaking, perceptions. Starch is also converted to maltose in the presence of enzyme phthalate, and these bacteria are killed by the lysozyme which are present inside the mouth. So these are some of the functions related to salivary juice. Next comes that is the gastric gland, and the secretion will be gastric juice. So gastric glands are mainly associated with the stomach now. So there are around 35 million gastric glands which are associated with the stomach, and there are two different regions. Anterior part is called as cardiac region, middle portion is called as fundus region or fundic region. So in the anterior part, the cells present, they are mainly having mucous neck cells which secrete mucus. And middle part, that is fundic or fundus region. So these mainly gastric glands in the human have the four distinct types. Again, they are subdivided into four different types of cells. So peptic cells, or it is also called as zymogenic cells. So they secrete different types of enzymes or proenzymes, means inactive form of enzymes. They are pepsinogen and prorenin. And also they are having the production of gastric lipase which are related to the fat digestion. So lipase means what? Fat digesting enzyme. Next, you are having the oxynitic cells. So oxynitic cells, they mainly secrete HCl, and it is also called as castle intrinsic factor which helps in the absorption of vitamin B12. So B complex mainly it is controlled, and hyperacidity is the normal high degree of acidity due to secretion of large quantity of HCl in gastric juice. Next, you are having mucus cells. So they secrete alkaline mucus, urgent of. They are responsible for secretion or vasocontraction of serotonin and vasodilator histamine. That means they play very important role in the muscular movement. And the third one, we are having the posterior part. Here the pyloric region which will be having some gastrin or gastric cells which secrete hormone gastrin, and they are increased by the motility of gastric wall and stimulates the gastric gland for the active secretion. So this is the main function of the posterior part. So here you can see how the three different regions. So three different regions are the cardiac region, fundus region and pyloric region in this picture. And later how they are divided. So the muscular layer is present, longitudinal muscular layer is also present, serosa is the outermost layer, lumen is the innermost layer, and there are different types of layers like you can find rug region and also oblique muscular region. They all are present. And later these gastric glands, when you take a section, you can find gastric pits, gastric glands which are associated and they carry the function. So these all are the region. And later muscularis mucosa producing blood vessels and muscle layers. So this is the picture which explains you how gastric glands are arranged. And what are the functions of these gastric glands? So mainly they help in storage of food, churning of food. Gastric glands secrete gastric juice. So we should know about the gastric juice also. So it will be secreted around 1 to 1.5 per liter. Chemical nature, highly acidic. pH is 1.1 to 3.5 because it contains high amount of HCl, hence it is completely acidic in nature. Control of secretion. So here it is regulated by the gastrin hormone. Secretion, they are of three types. So cephalic phase, gastric phase and intestinal phase. So chemical composition, same thing, 99% of water, but inorganic salts are much contained, and HCl is 0.5 concentrated, that is castle intrinsic factor is also present. Enzymes, prorenin, pepsinogen and gastric lipase, they are differentiated. So what is the function of this now? So hence we know the gastric juice is acidic in nature. So it makes the acidic and also the action of gastric enzyme. HCl kills the microorganism and it acts as antiseptic and preservative. So once the HCl is functioned, so it has got more functions as activation which are kept in inactive form. For example, pepsinogen. It is also an inactive form which will be activated in the presence of HCl, that is it will be converted into pepsin, that is active form. Next, prorenin is converted to renin or the active form renin in the presence of HCl. Later, proteins and peptones, they are converted into polypeptides and oligopeptides in the presence of pepsin. So casein, we all know that casein is the milk protein. So casein is converted to paracaseinate in the presence of renin and calcium. So lipids, they are converted into triglycerides and monoglycerides in the presence of lipase. So intestinal glands, so these glands in mammals, they are collectively called as crypts of Lieberkühn. You can see how the digestive gland, they are separated and form different types of layers. And all the part of small intestines are related to secretion of intestinal juice, which is also called as succus entericus. And succus entericus, it will be secreted around 1.5 liters per day. Chemical nature is alkaline. pH is 0.6 to 8.3 and controls the secretion of nervous and hormonal secretions also. Chemical composition, 99% of water, mucus is present, inorganic salts and enzymes. So this is the structure how you can find the small villi are also arranged in the small intestine. So these are the villi which helps in the absorption and the regions from stomach to how it is changing from one region to another region. So this is about the gastric glands. So you can find the villus, that is small villi also, which helps in absorption. So what is the function of these intestinal juice then? So intestinal juice, the main function is to inhibit the action of gastric enzyme. So it will stop the function of gastric enzyme and that leads to make the condition alkaline. Starch is converted to maltose, isomaltose and dextrin in the presence of maltase. So whenever AAC comes, remember they are enzymes. Isomaltose is converted into glucose in the presence of isomaltase. So maltose is converted is also same, but starch it will be converted in the presence of amylase. Lactose, it is converted in the form of glucose and glucose in the presence of lactase. Sucrose, sucrase. Polypeptides and oligopeptides, they are converted into amino acids in the form of the erythrin. So these are the some of the functions, and later the trypsinogen, which is in inactive form, they are converted into active forms of the enterokinase. Lipids, in the presence of lipase, it is converted to fatty acids, glycerol and monosaccharides. The phospholipids, they are converted into phosphorus, fatty acids, glycerol and monoglycerides in the presence of phospholipase. Organic phosphates, in the presence of phosphatase, they are also converted to free phosphates. Nucleic acids, they are converted to nucleoids in the presence of polynucleotidase, and later the nucleotidase, they are converted into nitrogenous bases. So next we are having the organ, that is, or the gland, which is the pancreas. So these pancreas are nothing but they are single endodermal structure and they are leaf like, which is yellowish in color. They are heterocrine gland mainly present in the ascending and descending limb of duodenum and which opens up in the duodenal region of the pancreatic duct. So they can be divided into exocrine and endocrine. So the first exocrine, that will be a larger part of around 99%, and which helps in the secretion of alkaline pancreatic juice, and the main duct is called as duct of Wirsung. And this duodenal region which passes through the hepatopancreatic ampulla. And this hepatopancreatic ampulla, it has got the accessory pancreatic duct named as duct of Santoni. So this is about the exocrine. Endocrine is very much important. So it is the minor part, but it consists of islets of Langerhans, and which consist of 4 different types of cells, that is alpha, beta, gamma, delta. Sorry, gamma is not there, delta and PP cells. So the function of these alpha cells is to secrete the glucagon, beta is to secrete insulin, delta is to secrete somatostalin, PP is to secrete pancreatic polypeptide hormone which controls somatostalin, and this secretion passes directly into the blood. So about the pancreatic juice, which will be secreted around 1 to 1.5 per liter per day, and chemical nature alkaline, pH will be 7.1 to 8.2. Control of secretion mainly hormonal and neural. Secretin hormone stimulation, the production will be higher. Chemical composition 99% of water, enzymes and salts. So what is the function of this pancreas and the enzyme? So the islets of Langerhans which secrete the insulin and glucagon, they are the main function. The exocrine part of pancreas, they secrete pancreatic juice. So first elastate, that is, it is a elastin protein. So now if you remember the trypsinogen, which is active form of trypsin, so that trypsinogen in the presence of enterokinase, it is again converted to trypsin. So trypsinogen is converted to trypsin here because it has to be activated form, trypsinogen. Then chymotrypsinogen is converted into chymotrypsin in the presence of trypsin. Polypeptides and peptones, they are converted into tripeptides, dipeptides and oligopeptides in the presence of trypsin. So starch is converted into maltose, isomaltose and the dextrin. So emulsified lipids, they are mainly converted into fatty acids, and at last you will be having nucleic acids, nucleotides and polypeptides which are converted into their respective smaller objects like nucleotides, nitrogenous bases and oligopeptides. So next moving to a very large organ, that is liver. So liver is the largest and heaviest gland of the human body, and it is divided into 2 different lobes by falciform ligament. So falciform ligament, it separates the liver in 2 forms, that is right lobe and left lobe. So it is a sac structure where gallbladder is attached, and it has got a common hepatic duct, and this common hepatic duct later it passes through the common bile duct where the bile juice will be secreted, and the main pancreatic duct from hepatopancreatic ampulla, that opens and it is guarded by the sphincter of Oddi. The basic structure we know that it is the hepatic lobule, so they play a very important role in the liver cancer and all, because they are the structure and functional unit of the liver, and this thin connective tissue is called as the Gleason's capsule. And later on the central vein and cord like manner, and in the form of phagocytic cover cells, that what happens, they mainly fight against the bacteria or they eat the bacteria itself. So functions of liver, it has got n number of functions like mainly detoxification, and worn out erythrocytes are replaced by the liver cells only, and it also helps in the fluid containing bile salts, bile pigment, cholesterol. So these all are mainly controlled by the liver, and also it helps in the production of or conversion into glycogen, that is glycogenesis, glycogeogenesis and glyconeogenesis. These are the very important terminologies which can be asked for 2 marks. So under abnormal condition, liver can convert protein and fat into glucose, and later new sugar will be formed. So that new sugar is called as gluconeogenesis. Neo means new. So these are some of the vitamins which I have mentioned about the vitamin A, and also there are blood clotting factor which are associated with the liver production, that is fibrinogen and prothrombin, and it is also called as site of detoxification. Okay, coming to last gland, that is gallbladder. So gallbladder, it is a smaller, somewhat bluish in nature and pear shaped, and it has got the connection with the liver. So the production of bile is it is around 800 ml per day, because bile is stored inside the gallbladder. So some secretion about the hepatic cells. Storage site will be the gallbladder. Color is somewhat greenish. And what are the functions of this bile? It mainly helps in emulsification of fat, means the digestion of the lipids, that is emulsification. And the main thing, that is it increases the alkalinity, and the medium suitable for the enzyme actions will takes place. And stericobillin and urobilin are mainly two important formation by the bilirubin and biliverdin, and produce coloration in fecus and also the urine. So this is about the digestive glands. So already in this chapter we have covered gross anatomy of human, that is including organs and glands, and also I have explained about how the digestion takes place in different parts. You can relate it with the protein, or you can relate it with the carbohydrates, or you can relate it with the lipids, because you know in which part what type of digestion takes place. In next class we will be discussing about the hormonal control and ruminant digestion. So that's all about this session. So thank you.